Australia is one of the world's biodiversity hotspots, but documenting that diversity is fundamental to our knowledge and to science in general, because we need to know how many species we have in order to document that diversity properly. And this goes both on a molecular level by looking at DNA, or on a morphological level by looking at morphological characters that can individually define species. But we don't just use morphology. Geographic characters or geographic differences between individuals is also relevant to differentiating between different species. The pea family, or legumes, are probably one of the most diverse families of plants throughout the world. And Australia is certainly no exception. We have exceptional biodiversity of legume flora here in Australia. And because of that diversity, it's very important that we understand and we categorise the diversity and understand how many species we've got here in Australia. The plant next to me just here, this is Platylobium, and Platylobium is in the legume family here in Australia. Platylobium is a really nice and interesting plant, and it's a member of a group called Mobileae, which is nested within the legume family. This is a really diverse group consisting of a lot of species, and if you think the typical pea-flowered legumes, this is what you'll get from this group of Mobileae. And this is what the focus on our current research is. What we're aiming to do is to understand the genetic and morphological diversity of this group, so we can find out how many species we've got here in Australia and what is the diversity like between these species and how many new species do we potentially have to science.